Many of you all may be familiar with famous pandemics that have affected human populations, from the Black Plague to the recent COVID-19 pandemic. But a disease that you may not be familiar with is chytridomycosis. Instead of affecting humans, this disease causes cardiac arrest and suffocation in millions of amphibians worldwide. Chytridomycosis is an amphibian skin disease caused by the fungus Batrachotrydium dendrobatidis, which is often called BD for short, that has been the cause of worldwide amphibian declines in the past few decades. In this presentation, I will break down the origins of this fungus, the biology of the infection, as well as conservation efforts to save amphibians before even more species become extinct. First, to understand the scope of the disease in amphibians, you must first understand its origins and its spread throughout the globe. So just like how COVID-19 was first discovered in China, chytridomycosis was first discovered in Queensland, Australia in 1993. This was just the beginning though. In the past 30 years, this disease has become widespread in all continents except for Antarctica. This is huge. From an article by Dr. Fisher and his colleagues published in 2009, BD infects over 350 amphibian species and is driving the decline of over 200 of these species. That's almost 60% of species that are disappearing. So now you may wonder, why does this disease spread so fast? Well, in a review article by Dr. Skarad and their colleagues in 2007, they tell us that in addition to infecting a huge number of species and being extremely deadly, this fungus can also persist at really low densities in the environment. So when BD enters the environment, it is especially difficult to get it out and recover the populations of amphibians that have been affected. So now that we have a better picture of the spread of the infection, let's dive a little bit deeper into how the fungus actually affects amphibians. So according to Dr. Rosenblum and their colleagues in 2010, this fungus, BD, lives in the water in something called a motile phase. This basically means that the individual fungi cells have a motor on the back of it called a flagellum that propels them like a motorboat through the water. The fungus uses this tiny motor to swim into the frog skin to start their colonization. They enter the outer layer of their skin called the keratin layer of the epidermis, and they start multiplying rapidly like a photocopier. These new spores are then released back into the water to start the cycle all over again and infect new amphibians, leaving the frog with the disease called chytridomycosis. So you may be wondering, what's wrong with just a simple skin infection? You and I get things like rashes and survive just fine. But the issue is that amphibian skin does a whole lot more than our skin does. According to Dr. Voiles and his colleagues in an article published in 2009, not only does their skin protect them from the external environment, it is also involved with the exchange of respiratory gases, water, and electrolytes, the stuff you get in Gatorade that keeps you hydrated. So the skin is kind of like our lungs and our stomach in this sense. However, when the fungus infects their skin, the amphibians cannot maintain these functions at all. In a different study by Dr. Voiles and his colleagues in 2007, they saw that the amphibian skin, after being infected, causes them to become suffocated to death and die from cardiac arrest. After understanding how devastating this infection is to amphibians worldwide, conservation strategies are crucial in preventing even more amphibian species from becoming extinct. There are two overarching approaches that show themselves in three specific management strategies described by Dr. Scheel and their colleagues in 2014. These approaches are number one, reducing the fungus in the environment or on the amphibian itself. This can be done by environmental manipulation. For example, you can increase heat sources because the fungus survives better in the cold, which reduces the overall amount of infection and the amount of fungus present in the environment. The second strategy is to increase the capability of populations to survive despite this increased death and disease. This can be done by reintroducing amphibians back into the population that chytridiomycosis has wiped them out. Or you can do this by selectively choosing amphibians to procreate who are resistant to the infections. So overall, these strategies can be combined and tailored to the specific location of the outbreak, as the species and environment can differ considerably. For instance, raising water temperatures through heaters is much more useful in the winters of Indiana versus in the tropics of Costa Rica. Scientists are trying to fight this battle from all fronts, so they are figuring out the best combination of approaches to save frog species before their extinction. Chytridiomycosis, caused by BD, has devastated amphibian communities worldwide, all within the past 30 years. This presentation has covered the global emergence of the fungus BD, its life cycle in infected waters, the resulting amphibian infection chytridiomycosis, and most importantly, conservation efforts to help prevent the infection. Overall, research into this emerging infection is crucial in the protection of amphibians before more species are gone for good. Thank you.